Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome, everybody, again to another episode of our show, bringing you another really awesome guest today involved in creating a better tomorrow on some really unique fronts. Today, we have the honor of being joined by Dr. Sofiya Milman, who is Associate Professor of Medicine in the Division of Endocrinology and Geriatrics and Director of the Human Longevity Studies at Einstein's Institute for Aging Research, Albert Einstein College of Medicine. Dr. Milman's research focuses on a really unique population of centenarians and their families who are generally free of age-related diseases, which demonstrate extreme healthy longevity running in these families, ultimately suggesting a heritable basis for the phenomena. And her team is involved in conducting a range of translational research projects focused on the discovery of the genomic mechanisms that help regulate metabolic pathways, endocrine dynamics, all that potentially could be involved in protecting these people against age-related diseases, including diabetes, Alzheimer's, cardiovascular disease, and so forth. Dr. Milman became really fascinated by both physiology and genetics during her college years, later on at SUNY Stonybrook School of Medicine, where she received her medical degree, and then following an internal medicine residency and fellowship in endocrinology at Montefiore, the opportunity arose to combine these passions. She began working with Dr. Nir Barzilai, who we recently profiled on the show, and then during her a couple of years of postdoc training in translational gerontology and genetics at Einstein's Institute, really got hooked on the subject, and it became her life's work. A lot of really interesting themes to get into today. Excited to have her with us. Dr. Sofiya Milman, thank you so much for taking time to come on the show today.
Thanks. Happy to be with you.
I would love to start off, Sofiya, by sort of, before we get into all things aging and gerontology and centenarians, love to start off with you and a little bit of your background in endocrinology, because I was taking a look at some of your early publications, sort of pre-longevity, and you were involved in publishing on everything from hypothyroidism, complications of prediabetes, type 1 diabetes. What got you into endocrinology to begin with? Love to hear that story.
So I got interested in endocrinology, I think, for several reasons. One is because I love physiology and the complexity of the endocrine pathways and the endocrine loops and the way one thing leads to another really fascinated me. It's a very cerebral area of medicine where you can just kind of sit back and figure things out just by knowing what normal physiology is. So that was very appealing to me. Also, I felt that endocrinology is very systemic, right? The hormones circulate throughout the entire body. It affects all organ systems. So it was an opportunity to, although subspecialized, still kind of think of the whole body when treating patients. And also was an opportunity for prevention because I toyed with the idea of becoming a cardiologist at one point, but I figured if I become an endocrinologist, I can probably prevent a lot of the cardiac complications that one sees as a cardiologist. And so it was really about, getting ahead of the game.
And connecting, because I said you you're doing a lot of work in these different areas of endocrinology. Longevity starts showing up in your sort of research path. And well, I guess you're known at the time for the work that you do in IGF-1, insulin like growth factor 1. You also start looking at things early on in terms of longevity connected to various aspects of lipoprotein levels. Related to things like vitamin D insufficiency. And it was interesting, when I was reading sort of through your bio and some of the work you were doing, one of the things that you run into when studying these various hormonal pathways is it's kind of difficult at first to understand, is a hormone causing aging? Is it preventing aging? Is it protective? Is it somewhere between? Because these hormones have these interesting dual effects, antagonistic, pleiotropy, and so forth. Talk a little bit about that, if you would, and a little bit about what you experienced early on in studying things like IGF-1 and sort of the dual nature of how to understand something like a specific hormone connected to something as complex as longevity.
Yeah, so I think it's a really important question that you raise, right? How do we decipher between associations, right, and causations? And I think that's a really, really important aspect of any research that one does, because a lot of research is really just associations, right? Looking at an individual at one point in time and, let's say, measuring a hormone or some other biomarker and saying, well, the people who have this biomarker, right, higher levels of this biomarker or lower levels of this biomarker, are also more likely to have disease X, Y, or Z, right? But those are just associations, because you don't know whether the hormonal level actually contributed to the disease, right? Or maybe it was a response to that disease, right? Maybe the disease came first, and what you're seeing in your hormonal milieu or in your biomarker level is actually caused by that disease, right? The disease caused the hormonal level to change. Or is it totally unrelated? They just happen to live together. Right? There's a classical example that is always taught early on in epidemiology, that there was a paper that came out that suggested that drinking coffee was associated with cancer. And what the researchers had failed to appreciate at the time is that a lot of coffee drinkers also smoked. And it was actually the tobacco, right, that caused the cancer. But if you don't ask the right kind of questions, and they said, oh, well, it's coffee drinking that's associated with cancer. And so we have to be very careful to make sure we pick up on a lot of these potential, what we call confounders, right, other confounding factors that go along with it. So we first started, a lot of our work had been, cross-sectional, meaning at one point in time, it was just purely association studies. But in order to really get at causation and to appreciate, is it the cause or the effect, one needs to look at things over time. Okay. And so we have embarked on longitudinal studies, right, or looking at things over time. And one way we do it is we measure, let's say, a hormone level at one point in time, and then look into the future and say, does a hormone today predict the onset of a disease 5-10 years later? So that could be one way to now get away from these pure associations, but look into the future and see what happens. And then, of course, ultimately, the way you kind of, put the nail on, kind of really seal the deal, right, is to do it either through animal studies, where you say, I'm actually going to perturb a certain parameter, right? I'm actually going to make a change, let's say, in that hormone level and see what the effect is going to be. But not everything from animals translates to humans. And so ultimately what you would need to do is, if you have a strong hypothesis, is say, okay, let me now do a placebo-controlled study in humans. And by altering, again, let's go back to the hormones, the level of a particular hormone, let's see if that prevents a disease or or maybe causes the disease. So really, you come from this kind of supporting evidence that often starts with cross-sectional studies, just observations and associations, to looking at things longitudinally as predictors. But I think at the end of the day, to really know if something is causative or not, you need a placebo-controlled study of humans.
And going now one step further, and then we'll get into centenarians in a moment. But so we have the, let's say, so the hormone or the specific moiety, let's say, now longitudinally looking at that. And now there's another aspect to your work, and again, I was really intrigued by the number of papers that you were involved in that now talk about sort of the complex nature of everything else going on, because you're involved in the recent the whisk-array, the plasma proteome profiling, where hey, we got lots of proteins here in the plasma. You've done studies on microRNA profiles of blood, complex combinations of inflammatory biomarkers. Say a few words, if you would, because I know it's tough, but nonetheless, these hormones and other biofactors exist in a very complex milieu. How has that affected your work as you continue to study everything that's going on in these patients? And we'll get to patients in a bit. When you have lots of things in that plasma or other biomaterial, fluid, whatever it may be, how does that increase, the complexity of what you think about on a daily basis?
Yeah, I think the human body is extraordinarily complex. And when we do our experiments, we really do it with a reductionist approach, right? We're like, oh, let's change one thing and see what happens. Whereas, as you point out, there are hundreds of thousands of things maybe perturbed. Yeah. So I think you do it one step at a time, with the recognition that things are very complex. I think it might be easier to do in animal studies, but even then, my colleagues like to point out, you have mice that are essentially genetically identical, and yet they have different outcomes. And so how much more complexity is there in humans when we are very, very genetically diverse and are exposed to different environments? So I'll kind of tell you maybe an example of how we came to appreciate this complexity and how we've been trying to tease it out. So at first, we did a study where we found an association between a low IGF-1 level of growth hormone, low growth hormone level, right, and longevity and better cognitive function. Okay, it was still an association in very, very old individuals, right, people who are 90 and older. So then we said, let's see if the same thing applies to younger individuals, and also if that same paradigm it holds up longitudinally over time. So we took a group of middle-aged adults, 65 and older, measured that growth hormone IGF-1 at baseline, and said, let's follow them out, right, 5-10 years, and see if there's an association, and found an association. Now, in the meantime, in the literature, there were a lot of papers published that didn't find such associations or such correlations. In fact, people have found associations in the opposite direction, suggesting that maybe higher levels may be protective, whereas we found that lower levels are protective. Or people have found no relationship whatsoever between the hormones and disease. And so that led us to thinking, why would that be? Why would people find different things? And so we began to appreciate, I think, two aspects. One is age, right? That age may actually matter. Quite a while ago, researchers began to appreciate that, children are not small adults, right? And I think now we need to appreciate that, older individuals, are not just chronologically older young individuals. There are significant physiologic changes that occur. And this includes hormones. We know that hormone levels vary with age.
Mm-hmm.
So growth hormone levels go down as we age. For example, that's one good example. Menopause, right, is another great example. I think that everybody recognizes that the estrogen levels decline post-menopause. And so we really need to appreciate, okay, how does the aging body change? And are the things that we know about, a healthy 20, 30-year-old still apply, right, to someone who's 70 or 80? And once we began to appreciate that, we said, okay, well, let's look again, right? Let's look at the literature and let's look at the data and see if maybe age is a really important component that may explain the discrepancy between the associations of this growth hormone, IGF-1, and disease.
Mm-hmm.
And that's what we actually found. We found that when we look at older age adults, like, above 50, I would say, so 50, 60, 70, 80, having lower levels of growth hormone IGF-1 has actually been shown to be protective against multiple age-related diseases, cognitive decline, diabetes, heart disease. But if you look at people who are younger, they actually benefit from having, higher levels.
Yep.
They could actually be maybe harmed. We don't know yet, but they don't do as well, at least from epidemiologic literature, with lower levels.
Mm-hmm.
So there seems to be a transition, as we age, when something that might have been good for you when you're young may no longer be good for you as you get older.
Yep. And
we have been able to show that very clearly through our research by actually looking at individuals at different age groups and appreciating that the associations between and the risk between the hormone levels and disease actually varies by age. And the studies that did not appreciate any association lumped everybody together. They took 18-year-olds, they took everyone between age 18 and age, let's say 120, and pooled them all together with the expectation that the effect will be exactly the same, irrespective of your age. And we're actually finding that you have very different effects based on one's age. So that was one, I think, complexity that we appreciated and we're actually able to show.
Mm-hmm.
The other aspect is, right, what impact does disease itself have on measurements of our markers? So, for example, something like growth hormone, right, which promotes growth, as the name suggests, is really a marker of, everything considered equal, right, assuming there's no underlying disease. The growth hormone is really considered a marker of, good health. When you're healthy, when you have sufficient energy sources, then you can grow. Now, if you're not healthy or you don't have enough energy, right, external energy sources coming in, you're not going to grow. Your body's not going to put in energy into growth when you don't have enough calories, let's say, coming in, right? Because you need to conserve that energy for other more important life-sustaining processes, like maintenance of, cell homeostasis, right? And just day-to-day kind of housekeeping, right? Not grow and expand. Same thing with reproduction, right? You don't want to reproduce when you have limited resources.
Yep.
So if one it doesn't have enough access to nutrition, let's say, right, or resources, or is ill, like let's say with cancer, right?
Right.
The growth hormone levels are going to go down because the body is really not interested in maintaining growth. It needs to focus on the task at hand, maintenance, and day-to-day activity. So if you just take people and random, right, and you measure their growth hormone and you say, oh, people who have low growth hormone levels also happen to have more heart disease, more cancer, they're weaker.
Yep.
Well, what you might actually be appreciating is the fact that, well, that disease has caused their levels to go down.
Mm-hmm.
Okay. And so you really have to tease apart, is it the chicken or the egg? What came first, what came second, what's the cause, what's the effect? And again, the way to do it is to look longitudinally, because if the person has been healthy and their growth hormone levels have always been low and they continue to be healthy, well, then you know it's not causative. But if you see someone's levels were average or high, right, and then they got sick and their levels plummeted, well, that's an indication that that low level was a result of disease. And you really need to tease those things apart when you look at large populations, and we're trying to do that.
And speaking of large populations, you are the principal investigator of the superagers family study, and then again, Nir introduced us a little bit to the topic, but it's really you that's leading it to sort of on steroids to potentially recruit 10,000, I believe, of these superagers and their children, maybe up there themselves. Talk a little bit, just if you would, about the dynamics of well, you can reintroduce us to it, but also the dynamics of expanding something like this. Interesting facts, challenges. I mean, I'm sitting here actually in Philly, and I read you were in the paper here just a few weeks ago about one of the people you recruited locally. It was a 96-year-old geneticist. So any interesting stories about things you're running into as you're recruiting these fascinating people? Please take some time with that.
Sure. So the premise behind what we're doing is really to try and identify what are the biological drivers of healthy aging and longevity, right? The people who do live to be 95 and older are really quite rare in our population. And what's even more, I think, surprising about a lot of these people is that they're relatively healthy.
Yeah.
So that tells us that the human body is capable of living long, but more importantly, healthy lives. And if it's within our human biology to do so, then our ultimate goal is to expand that opportunity to everyone, right? Because right now it's the very few select individuals who enjoy those long, healthy lives. And in order to do that, in order to be able to extend that opportunity to others, we need to understand how these people were able, right, to live long and healthy lives. And there are numerous studies, not too many, but a few, across the country and across the world that have been specifically enrolling older individuals, either those age 90 or older, 95 or older, 100 and older, depending on the study design, in order to understand what it is that's unique about them. And so far, a lot of evidence suggests that genetics play a significant role in contributing to healthy longevity. And for people who are living to these extreme ages, genetics become even more important, and I think for other populations who don't live as long. And so the goal that we have for the superagers family study is to enroll, as you said, 10,000 individuals age 95 and older and their family members, with the aim of ultimately identifying genes that contribute to their healthy longevity. The reason we want to enroll so many people is because you really need a large population to study when you want to identify rare genes, right? Genes that are rare in the population that contribute to this very extreme phenotype, right, of being 95 and older. And we do have several cohorts now, but they're not big enough. So we really need the 10,000 to be able to make those discoveries. And once we make those discoveries of the genes, we're not really looking at genetic therapies necessarily, right? What we really want to understand is what is the function of those genes, right? How are they altering hormones or proteins? How are they impacting the biological pathways, the physiology of the human body? And once we understand what are the physiologic differences between people who have those protective genes and people who don't, then we hope to be able to design medicines based on that knowledge that we can give to people, right, who did not win that genetic lottery, who do not have those longevity genes, so that what we learn from this small elite group of individuals can actually impact the broader society and the healthcare system.
And one other part of it, I believe that I read that, in addition to the genes, you're also looking at microbiome, I think, some dietary. Is that also part of, specifically in the superager study, or is it just genetic? I'm gonna make sure I'm thinking of the right study.
Yeah, so the superager study is primarily focused on genetics and associating that with individuals' clinical history. We intentionally made it very easy and very simple because we want to enroll a lot of people. So we really want to be very inviting, and we don't want to turn people away because of increased burden. So it's actually a remote study where people can sign up anywhere from the United States. You can go to our study website at superagerstudy.org, and they can register, learn more about the study. It involves answering some questions about their overall health and their medical history online. And then we mail a saliva kit to the individual's homes.
Cool.
And they just send the kit back to us with a little bit of saliva that we then use to extract the DNA for this research. Everything is done under very strict regulatory practices. We're very serious about maintaining people's privacy, both for the genetic and the medical information. But ultimately, you can do the entire study, you can participate in the study completely from the comfort of your home. And that's very, very intentional in order to make things easy and to enroll as many people as we can.
Excellent. And I was just, one thing was kind of interesting, just thinking about my own family. We don't have any centenarians, but we have some late octogenarians, early nonagenarians. And when I think about, a lot of the work that you've done in your group, which, talks about sort of the compression of morbidity, I think about the folks in my world. They didn't compress their morbidity. They got cancer and heart disease and all that stuff, but they somehow survived. They sort of got through it. And I was very intrigued. There's a very recent paper that you published, I think just a couple of days ago, actually, on frailty resilience, and sort of, when I think of resilience, I think of not, I have not prevented something from happening to me, but I have bounced back from it. And I was just interested, and maybe I inappropriately sort of separate resilience and robustness as sort of two different things. But where does resilience fit into this? Are these sort of people only with they've lived long and disease hasn't happened, or are they what I see in my immediate landscape of, no, they're getting sick, but they have the ability to bounce back? And I think of them as two different things, but can you just say a couple words about resilience in the picture here?
So first, let me tell you that your family members are welcome to enroll in the Superagers Family Study because we enroll people 95 and older. So if they qualify age-wise.
Not there yet, but close.
Okay. So I think what you're bringing up is an important point. So there are people that completely escape, from all disease. Yeah. So they're, say, 100 and have never had any diseases that we associate with older age at all. No cancer, no Alzheimer's, no diabetes, no high blood pressure. And then there are people who had developed them and despite that, have survived. So a colleague of mine actually terms them survivors. So they're escapers who never had it, and then there are survivors. And there's probably something about those survivors that's unique, and I guess you could call it resilience. But so the way we kind of define resilience is doing well, or overcoming some sort of adversity. One example may be, somebody had a disease, but they were able to bounce back and recover, and continue to do well. Another way we think of resilience is that people may have had genetic risk. They may have been genetically at risk for a certain disease. So they carry genes, let's say, that put them at risk for Alzheimer's disease. And, everyone else with those genes would have developed Alzheimer's disease by the time, they're 70. And yet these unique individuals maintain normal cognitive function and have no signs of Alzheimer's disease when they're 90. So they are somehow able to be resilient to their inherent risk factors. which is fascinating. And that's another thing that's been quite remarkable, that there are quite a few studies that have now suggested that these centenarians that live long, relatively healthy lives don't always lack risk genes. Some of them may not have those risky genes for diseases, but many of them do carry the same risk genes for multiple diseases. And yet they never get those diseases. That means that there's something else about them. Maybe there are other protective genes that counteract the effect of disease-causing genes that allows them to be resilient.
Yeah. It's an interesting subpopulation, but I appreciate you giving your insight on that as well. So one of the things I did not mention in your bio is that in addition to you being an MD and having done all the postdoc work, you also have a master's in science in clinical investigation. And I thought, a very interesting, in addition to all the research publications you've been doing, you've published as well, sort of on the unique nature and challenges of running clinical trials in this space. The 2016 Strategies and Challenges in Clinical Trials Targeting Human Aging, and Nir was there, and Steve Ostad also. And both of them, we chatted a bit when I did the shows on about TAME and metformin. Would love your perspective, as you think about, okay, we're going to do the SuperAger study, and I'm going to find out about gene A, B, and C, some great targets there that are novel and maybe come with some unique mechanisms. What do you want to see as an expert in clinical sort of investigation design as well? These health span geroscience interventions look like? Because I know there's a couple different schools of thought out there now as we sit in 2023 that maybe we should only target diseases of aging and then think about broader health span trials later on. Or do we start, and are we aggressive with, setting up a health span study in the short term and don't waste all the time targeting specific diseases, what pharma's been doing. What would you like to see? If the sky opened up and gave you all the money you needed to do this stuff, what model do you like? And where do you think this all should be going?
Yeah, it's an interesting question. I think a lot of the things that we are seeing now on the ground are really dictated, right, by the realities that we're dealing with, the regulatory realities and financial realities. I think the science has come a long way where we do have enough evidence, at least from preclinical studies, right, from animal studies, that there are certain pathways that if you target them, you can impact more than one outcome, right? You can have an impact on more than one disease. And these are these classical aging pathways. So if you intervene, you could have more than one beneficial effect, both on function as well as delay or prevention of multiple diseases. So the evidence is there. So ideally, I would like to see us translate that into clinical trials, taking some of the more promising compounds that target the well-studied pathways and see if we can have that intervention in humans. Now, the challenges are clear, right? And I'm sure you've had many others talk about them, where to do such a trial would require a very long time. You might need a lot of people, a lot of resources. So kind of a shortcut is to say, okay, let's take one of these gerotherapeutics that do target the aging pathway, but rather than looking at multiple outcomes, let's look at one. And that's what I think a lot of people have embraced more recently. I think if we are going to do that approach, then we better also measure other aspects. And I think people are, right? So if I'm going to take a gerotherapeutic and say, well, I'm going to focus on one particular disease and see if this gerotherapeutic prevents it, let's say diabetes, right? Okay, that's fine. That could be your primary outcome, but make sure you're also measuring, other potential secondary outcomes, which are all the other aging diseases. So at least you have some data. You are collecting that data, and maybe that's not your primary outcome, and it doesn't hold as much weight, but at least there's data there, to drive more hypotheses to back things up so that in the future, a more, aging focused trial can be done.
And one other thing, Sofiya, while I have you, I just have to ask because it sort of the theme of sleep and sleep hygiene is so sort of exciting nowadays, or talked about everywhere. Can you just say a few words about your December 2017 study on sleep patterns and families with exceptional longevity? Because I'm a horrible sleeper. I haven't been a great sleeper in years, but I was kind of intrigued by the the insight from this paper. So if you could just say a couple words about what you learned about sleep as you were studying some of these patients.
I'll say one thing that we learned about sleep in that paper is that it's very difficult to study sleep, especially when you're asking people, right, to recall, their sleep patterns.
Right.
And it was interesting that I don't know that we necessarily walked away, with one specific takeaway, because there are people who, let's say, slept fewer hours during the night, but yet they napped during the day, probably to make up for it. So I don't know that, after doing that particular research project, that we, have the answer of what is the right or the best way to sleep. Might also be individualized. That being said, sleep is important. Sleep is restorative, and we should be trying to get sufficient sleep. I think many of us don't, myself included. But actually, there's one goal that I set for myself, to be healthier, and that was, to make sure I do get enough sleep. So I think sleep is fascinating because a lot of things happen when we sleep, right? There's a lot of restorative processes occur in the brain and in the body. So I think there's, a lot more to be learned about that.
Cool. Before I let you go, Sofiya, what's coming up for you as we get to the end of 2023, as we enter 2024? I mean, I notice you do, in addition to being in the lab and at Einstein, you do get out on the conference circuit, attending, a variety of different types of seminars and Alzheimer's and longevity and so forth. Any things we should know about, separate from the super agers recruitment, but that's happening in the coming months? Conferences, places that we can listen to you, potentially run into you, anything else on your schedule that I missed, please?
I guess nothing specific comes to mind, but I guess we're watching closely, the space of gerotherapeutics and looking for breakthroughs, because I think we're very close. I wouldn't be surprised, and in fact, I expect that in the next decade we do start seeing compounds that are effective and hopefully will be in clinical use. I would also say, I think what we've been really trying to do is to educate people more about geroscience and understanding that the biology of aging impacts every single system and organ system. And so I think educating our other clinical colleagues, right, in their own medical subspecialties about the role of aging physiology and appreciating that and appreciating the fact that it may alter other things, and that we really need to think of age in very physiologic terms and not just chronologic terms. So that's, I think, also been really important for us to kind of get the word out.
Yeah, no, it's so exciting. I mean, you have obviously in the centenarians, you have a very important population. You think quite broadly in terms of all these biologic targets, and you understand clinical development. So you're definitely at the epicenter of everything that's going on. And I'm a big fan of what's been happening there. I'm really honored that I had the chance to have you on to sort of take us into the future as far as the super agers program and where it's all headed. But wishing you the best with the programs, and hopefully we could do a follow-up as things continue to move forward next year. Again, for everybody that's going to be listening to this particular episode of our show across the various podcast networks or watching on our YouTube channel, you've been listening to Dr. Sofiya Milman, Associate Professor of Medicine, Division of Endocrinology and Geriatrics, and Director, Human Longevity Studies, Einstein's Institute for Aging Research, Albert Einstein College of Medicine. Sofiya, I want to thank you for taking the time out of your schedule to come talk to us about these topics and what you're up to. Obviously, thank you for what you do. And as we like to say on our show, thanks for helping to create a better tomorrow via the insight you're generating and programs. Really great story. And, thank you so much for sharing it with us.
Thank you, Ira. I enjoyed our conversation.